Hey, thanks everyone for joining. We'll go ahead and get started. So, today we're going to talk about soil biology and what's happening beneath the soil. I'm Dave Stark. I'm the co-founder and president of the agriculture business at Holganix. My doctorate is in molecular biology. Today we're going to talk a lot of science, and I hope it's not too overwhelming. I'm going to try to keep it interesting and lighter. But for me personally, I'm a scientist by training. I don't really like to be sold. I like to know why things work and why I should do certain things. So that's what I'm going to try to do today. Is just really show you, the value of paying attention to the soil biology. So I've done this in the past, and I've had guys say, okay, so I've learned a lot, but I'm not quite sure what I'm supposed to do because I don't sell enough, and I'm still not going to oversell. But our product is called Bio 800 Plus. We have different versions of it, one for the spring that helps with germination, nutrient use efficiency, and such, one in the fall that helps with residue degradation. Actually, this slide, all of these pictures come from customers that have used our product, except that one at the very top. That's a dead nematode that's been killed by our product. That came from a researcher. So my point is the biology actually does a lot in your soil if we take care of it. So when I talk about biology, this is what I mean. And I've done this before, so I'm not going to, hammer a lot of the things I've done in other webinars, but there's a lot going on under the ground. So in a gram of soil, which is about the size of an M&M, a chocolate M&M, um, there are billions of individuals of bacteria, actinomycetes, fungi, protozoa, nematodes. I suppose I could add archaea, algae, etc. You get the point. But if you look at the biomass, that right-hand column, in the top 6 inches per acre, look at the massive range. There can be a couple tons of bacteria and actinomycetes. There can be almost 7 tons of fungi or 9 pounds. Now, we have done soil sampling now on about 1 million acres of farmland, and we've learned a lot from it. And I'll show you some of that in this talk. What we tend to see, though, is the way that we've been farming, and again, I'm no blame or anything, we tend to be on the low end of these ranges. So let's find out what's going on. So first of all, bacteria, archaea, actinomycetes, these are the ones that are the most abundant. I think of them as hungry teenage boys. They are the first to eat. They grab anything that's edible in the soil, and they're very important in that. So I talk a lot about fungi, but I don't want to overlook the role of these guys because they're critical. They actually perform a lot of the functions in the soil. They help a lot with nutrient cycling. They induce rooting. They help make the ground better. When they eat, they tend to release carbon as CO2. That's okay. Actually, most of these need oxygen, some do not. So how do we know? So I spent 36 years in Missouri, the Show Me State, and I like using pictures to kind of emphasize what I'm saying. So here in the upper left corner, that's fallow corn. So you've probably seen purple corn before. That's from low phosphorus. I've had customers that say I get purple corn every spring. I bet if you test that soil and you look at the total phosphorus pool, it's huge. So what's going on? Odds are there's not enough microbes to unlock that phosphorus to provide it to the plant. It's been tied up in the soil. We're going to talk about this. So I'll give you a little more detail around phosphorus. The one on the right, that's yellow corn. Again, I bet you test that soil, total nitrogen, probably a lot. Could be tied up in bacteria. We'll talk about that too. But again, visual evidence that soil biology does impact crop nutrition. Now, fungi, these are degraders. They are critically important for so many things, and we'll go through a bunch of that. They eat bacteria, they build soil structure, they're great at unlocking nutrients. You can see excuse me, all that white feathery material, that's the fungal hyphae, mycelia. You can see how tiny they can get between soil particles and such. So fungi are critically important in nutrient cycling in so many soil functions. Again, how do we know? Well, here's a picture. Now, we don't want this particular fungus. This is called fairy ring. It starts in the middle of that circle, and as it spreads outward, it's breaking down organic material and releasing nutrients. That's why the leading edge is darker green. It's releasing nitrogen. So again, visual evidence, microbes impact nutrients. The last, the protists and the nematodes. These are really the guardians of the soil microbiome. I don't talk as much about them, but maybe in future webinars I will, because these really control, they keep everything else in check. These are predators. They eat bacteria, fungi, they eat each other, they eat organic matter. By the way, most nematodes are actually good in the soil. In that lower right corner, that's a picture of a nematode eating another nematode. So these are really the balancers. They make sure that nothing gets out of whack, that all of these microbes, which are designed to work together, continue to work together. So there are things that happen, because I get the question all the time. How often do I need to keep adding a diverse biological like ours that has all of these microbes in it in one product? The soil biome always is changing. There are things that we do to change it. Tilling, we're going to talk about tilling. Overuse of fertilizers, we're going to talk about that. I won't talk as much though about fungicides, fumigants, things like winter. Um, Microbes are full of water, they freeze, it kills them, doesn't kill them evenly though. Flooding, um, most of what you want in the soil needs oxygen. 48 hours underwater, the oxygen's depleted, things are changing. You can really tell if it smells like a swamp when that water drains, then you know that soil has really changed and needs to be re-inoculated with your beneficial microbes. So that's what our product is. This is my sales slide. We have over 800 species, really several thousand species of bacteria, actinomycetes, archaea, algae, fungi, protozoa and amoeba, even beneficial nematodes, food sources to help them start to get to work as soon as you put them on the ground. So we add that life back to the soil. So you've heard me talk about nutrient use. And through the years, I've just noticed how I've treated the topic, where I've started off pretty soft because I am not trying to bash fertilizer by any means. So please don't hear that. At the same time, through the years, it's just really apparent. If you want to get the most out of your soil, you got to stop using so much fertilizer, depending where you are on the spectrum, because some of you are probably already very efficient. Generally speaking, though, the way we use fertilizer is really inefficient. A lot of your money is still sitting out there in the field, not helping the crop, or it's gone into the air or down into the river. So the microbes can help us do better. Now, this is an example of a lot of the slides where I'm not going to go through the chart by detail. I'm doing the pictures on purpose, and you can see the fine print. That's a scientific reference. This presentation is available to anyone who wants it. If you want to learn more about any particular subject, this will help steer you in the right direction. But I do have a few main points. Soil root exudates really help control the plant-soil feedback. Biochemically, the plant and the soil are always in communication, and the plant biochemically is determining its nutrient strategy. So, in a normal situation, the plant is farming microbes, actively attracting, actively repelling certain types of microbes to build the population it wants around it, because microbes mine the soil. However, when a plant senses that it has excess nitrogen or phosphorus, it slows down putting exudates into the soil. so it's not feeding the microbes as much. So in that the exudates are carbon, and I don't mean carbon like charcoal, whatever. I mean like carbohydrates, proteins, and fats. Food, they're fueling the microbes. If we don't fuel the microbes, you're going to feed the plant with fertilizer. So you have a choice. Do you want your microbes to feed the plant or your fertilizer dollar? That's what it comes down to. How do we know? So we're going to hit on this. So this is nitrogen use efficiency in corn, rice, and small grains. 230 published studies summarized here. If you choose to let your fertilizer dollar fertilize the crop and not the microbes, that's that box with the black around it. That what they found is you put nitrogen on, some of it the crop directly uptakes, a lot of it sits there or is lost. But in a biologically healthy soil, we get the situation on the right. Essentially, what this is the microbes consume the nitrogen. Most of your crops' nitrogen needs pass through a microbe first. This is good because they immobilize the nitrogen, they keep it in the root zone, more of it stays active, more of it ends up back to the crop. In red, you can see the bottom line that I think we all care about. Biologically healthy soils produce greater corn yields per unit of fertilizer input. That's what we want. So why? Let's dig in a little bit more. And if you've seen my webinars, you've seen this slide. So when we put fertilizer on the ground, which is usually some form of ammonia or nitrate, and even urea, which then gets broken down, microbes eat it. And it's the hungry teenage boys, the bacteria, actinomycetes, etc., that eat it first. And that's good. They live in the root zone. They don't erode as easy. They don't wash off as easy. They don't volatilize. They can lose some to the air, some nitrogen, but they keep it there. But it's bad. Remember the yellow corn? It's because these guys need a lot of nitrogen. For every nitrogen, there's 5 carbons. That's an incredibly high nitrogen requirement. But the way God designed this, their predators who eat these and keep them in check have a much lower nitrogen requirement. So fungi 15 to 1, but really it's the protists, the guardians of the soil, the protists, the amoeba, the nematodes. One of these can eat 10,000 bacteria a day. Their carbon to nitrogen ratio is 1 to 30. So when they eat bacteria, 10,000 a day, that much nitrogen is toxic. They have to get rid of it. So they spit it out in the form of ammonia nitrate, where in the root zone, because that's where the bacteria are, what form does your plant take up the best? Ammonia nitrate. And again, you probably heard me say this is not evolution. If you don't know, a molecular biologist is an expert in evolutionary theory, especially from the DNA and biochemistry standpoint. And I just tell you, before, it just didn't happen that way. If you'll indulge me, because again, I like proof, I'm just going to show you a simple math slide. So this is the mathematical odds of making a small protein. By the way, one protein is not life. It's estimated you need over 480 different proteins to make even the most simple cell. And of course, you need a lot more than one of each. But this is just the odds of making one protein. And I cheated for evolution because it made the math easy. It didn't matter. So we're going to start with the 20 amino acids. That's what builds the protein chains. Amino acids exist in right-handed or left-handed. Only left-handed can be in a protein. Right-handed kills the function. So they're all going to be L, and they're only going to come together in the right peptide bonds. They can come together a lot of different ways, but I'm cheating to make some math easier. So for every position, you have a 1 in 20 chance of getting it right, times 150 times. The odds come out to be 1, it'll happen once, out of a number followed by 196 zeros. Now to put it in perspective, you see those first 6 zeros, that's a million. Then you add the green, that's a billion. The purple, that's a trillion. So every zero gets to be a lot. Follow the gray across, that's 50 zeros. Mathematically, I am told that if you have the odds of 1 out of a number with 50 zeros, the term is absurdly impossible. See the red? That's 80 zeros. That's the estimated number of all of the atoms in the entire known universe. Look how many more zeros you need to make one protein. So, in all due respect, if you believe this is all random, you need to believe something else. It's mathematically not possible. So I go back to God's design. In God's design for nutrients, it's more than just the nitrogen cycle and carbon nitrogen ratios. I've talked about this before in other webinars. It's the rhizophagy cycle. When plants sense that there's a certain population and mix of microbes, it'll start absorbing microbes. Now remember, the microbe, the bacteria, actinomycete, fungi, whatever, has the same nutrient requirements as your crop. So if the plant eats the bacteria, it's getting all the nutrients that the bacteria has that the plant needs. Remember, it's being farmed by the plant because the microbe mines the soil. So most of the microbes die, releasing all the nutrients. Some live, they induce root hair formation, they go back out in the soil and remine it. How can you tell? We love doing root digs. You can find so much with a shovel. So if you look at the corn roots in the upper left corner, those three, they're covered in dirt. I couldn't zoom in anymore and make it the resolution really visible, but they're covered in root hairs. Look how clean the others are. No roots, very few root hairs. Which corn do you think is growing in soil that has diverse microbes? The one on the upper left, the one treated with our product. Same with nodulation in beans. You need the rhizophagy to take the rhizobia and the other microbes that live in the nodule. It's not just rhizobia. So the more microbes you have, the more we trigger that quorum sensing, the better nodulation we get. So by the way, the grower did this picture not to show off the difference in the roots so much, although we again encourage root digs, but just that his corn was taller and greener. But we encourage root digs because roots are so important. If you think about the soil within the root mass of a typical plant, only about 1% is explored by the roots. Most nutrients need to be in contact with the root to be taken up, or with a fungus associated with the root, like a mycorrhizal fungi. So the more roots, the more root hairs, the more beneficial fungi, the more soil explored, the better the nutrient use efficiency is. Plus, we're going to talk about carbon and soil structure and all. What happens with all those roots when they die? They break up the soil, they leave channels for air and water, and it's also the #1 source of organic matter in the soil. Far more than the residue we leave on top of the ground is the roots and the root exudates. So phosphorus and potassium, same deal as nitrogen. A little bit different dynamics because I'm not going to talk about phosphorus carbon ratios and such, but microbes are key at unlocking what's there. So let's look. Again, busy slide, but what happens? So we mine phosphorus and we make phosphorus fertilizer, which you put on the ground. If you look at the total pool, the retained phosphorus, it's huge. Why? Phosphorus is very reactive chemically. It's going to react with what are called cations, things that have a positive charge. Calcium, if the pH is more alkaline, iron or aluminum even, and you don't want that because that's aluminum toxicity under acidic conditions. So the phosphorus is chemically reacting with what's already in your ground and it's getting tied up. The soluble pool is small, but it's microbes that are critical in bridging that gap between what's called the non-living environment, that chemically bound phosphorus, versus the stuff that's available for what's living, like your crop. So the microbes are the key for phosphorus use efficiency. Here's another way of looking at it, and this is kind of an eye chart, but it'll be important when we talk about soil structure too. So clay particles are tiny. They have a negative charge. Organic matter tends to be negative charge. Organic matter can be anything. It can be big, like pieces of leaves or dead microbes, but it's usually pieces of dead microbes, carbohydrates, fats, and proteins. Phosphorus can bridge it with the help of metals, because phosphorus is also negatively charged. So metals are stabilizing this. It can build together. This is actually a good thing because it helps stabilize soil structure. But we want that phosphorus released. What microbes do, and a lot of that phosphorus, you can see kind of there in the middle on the left. It's the P with the calcium, iron, that's Fe or aluminum. It's the metal phosphorus. Microbes secrete acids that break it apart, enzymes that can break all this down, or iron chelators that release the phosphorus, making it plant available. So again, a lot of P in your soil. The plant can't do these things, microbes can. And actually, most of the available phosphorus pool, if we look at it, is organic. Just like with nitrogen, it's living in the microbe. Phosphorus, it's in the living and dead microbe. So we want the biology to keep these things available. I can do the same thing for potassium. Visual proof. Remember, I'm from Missouri. I live in Indiana now, but I like to see pictures and evidence. So this is NDVI imaging of a customer in South Dakota. This is his home quarter. So that kind of purple red area, that's his house and yard and such. So remember in NDVI, the darker the green, the healthier the vegetation. Yellow and red is not so good. He started using our product in 2019. Look at the difference as you go through the years. Through this period, he has gone from over a pound of nitrogen and a lot of P and K to just over half a pound of nitrogen per bushel of corn and essentially no P and K, a little bit with a pop-up fertilizer. So he is saving a ton of money on his fertilizer input. His yields are still 200 bushel an acre plus, and I think it's 23 or 24. He won the state soybean dryland yield contest. So less can be more. This is taken last month in Missouri. Look along the row. Can you see the difference in the color of the corn? It's a straight line. Our product on the left, not on the right. Which crop do you think is getting more nutrients? I think it's pretty easy. We also had a customer in Iowa. Part of his field, there's 30 acres contiguous field that this customer farms for his uncle. He puts our product on everything, but the co-op fertilizes his uncle's 30 acres. And using our product, he has cut to where this year he did not apply any fertilizer. And his uncle was like, why is your crop always so much better than mine? It's the same field. So we did a soil test. Now, this is a combustion test where we're looking at all the nutrients. Look at, by the way, where it says no fert, kind of the light green versus the full fert, his 30 acres and kind of that bluish purple, almost 50% more nitrogen, almost double the available phosphorus, higher potassium. Look at the pH is better. The organic matter is better. Actually, the pH in that 30-acre field is low enough. He's getting a lot of his phosphorus tied up with aluminum and iron. No fertilizer, but higher available nutrients. And if you look at the fertilizer equivalent in the other columns on the right, it's massive. That's real money. And then look at carbon, almost double the carbon. What is going on? So now we got to look at what puts carbon in the ground. And you know, if you hear us, we're starting to talk about carbon now. Again, a lot going on in this picture. We're going to make it real simple. What is carbon sequestration? It's taking CO2, that carbon, and putting it into the ground. And you know that plants use sunlight, water, and carbon dioxide and make sugars. That's photosynthesis. That's what they do. When we look at carbon in the ground, almost all of it enters the ground via roots and root exudates. It stays in the ground because of microbial activity. So it enters through roots, it stays because of microbes. Now there's inorganic carbon I'm not going to talk about. I could, it's chemistry. Sadly, a lot of the inorganic carbon that's been in our ground has been lost because of acidification. It's calcium carbonate and it gets lost to CO2. It can be put back. It's a slow process. That's not what I'm talking about. I'm talking about how we put carbon, which again is the fuel for the soil. Again, it's not charcoal, biochar, diamonds. It's carbohydrates, protein, and fats. It's fuel, food. So organic carbon, which is an important measure of your soil quality, it's derived from organic matter. Do you know almost around 80% of it are dead microbes? Microbes once again pop up to being huge for how our soil works. How microbes stabilize this is they help physically separate it, and we'll talk about aggregates, uh, chemical association with mineral. Remember back to phosphorus. I showed you this clay particle, the phosphorus, and the organic material. Well, you don't need the phosphorus to be mineral associated. It can bind directly or with a metal like magnesium or calcium, whatever. That's called mineral associated organic matter. When it's bound like that, it's stable. Microbes can't break it down anymore. But it's not permanent. And we'll talk about this on the next slide. Now, there's also some forms of carbon that are slower to degrade, that are just more stable. I can tell you, we used to think things like humic acid was stable and microbes could barely break it down. It doesn't look like that's really true. That there is no form of carbon outside of maybe literally a diamond that a microbe can't break down. So it's not a matter of being permanent, it's a matter of being persistent. This is how microbes keep carbon in the soil. What they do is they'll break down your leaf litter, the dead roots, the dead microbes. Some of that will stick to a clay particle. Some of it will be encapsulated in soil aggregate. When it's stuck, it's stable, but other things can knock it off and make it available to be eaten again. Things like freezing, drought, uh, microbes secreting acids and such, or something that's a stronger attractant to the clay particle that knocks it off and replaces it. That's okay. The form of the carbon will change. Microbes will eat it again. As it rains, they move deeper and deeper into the soil where there's less and less microbial activity. They're eaten again. The microbes die. They associate with clay particles. They become more and more stable, especially as we go below a foot. The bottom line is microbes are essential to stable organic matter and organic carbon in the soil. This is just another way of looking at it in terms of how the mineral associated organic matter is the last part that leaves soil. So that's a lot so far. Oh, and I've got time. Aren't you lucky? Like I said, we've done 1,000,000 acres of soil tests. There's a lot of things we look for. We do this at our expense because we sell soil health, um, and we measure it with you. So we've talked about the increases in organic matter, which is mostly carbon and organic carbon. Again, that's really important. We'll talk about why again. But also look at the top, bulk density. We want bulk density to go down. Why? Well, if you think about it, the more open your soil is, the more water and air goes in and gets held. Everything you want living in the soil needs water and air. It's also ground that's easier to work. So if you're putting anhydrous down in the fall and the row doesn't close because you've got nothing but big giant dirt clods, that's because you have too high of a bulk density. We need to fix that. How does that happen? Well, remember the susceptibility of soils to compaction is a result of biology. It's not your heavy equipment alone. It's the lack of biology. So what's going on? If we look at soil, and you remember the phosphorus, we've got those tiny clay particles. That's what soil is. They're tiny, tiny particles called microaggregates. Um, I like to use the analogy or the visual of two 5-gallon buckets, one with a really finely ground sand, one with golf balls. You tap them both down really firm. Now pour water on it. What happens with the finely ground sand? It sits on top. If you turn the bucket to the side, you get erosion. It slowly goes into the soil. What happens with the golf balls? That goes right in. What we want is to convert your soil from those fine little micro aggregates to macro aggregates. How does that happen? Microbes. Microbes secrete glue. They're exudates from the microbes and even from some roots that glue those little aggregates into bigger ones. Then the fungal hyphae, that white feathery material, surround it like the skin of a golf ball. Now it's protected. Can you compact a golf ball? No, because it's got strength and structure, just like a soil macro aggregate. Now that carbon that's inside your organic matter is protected from the hungry teenage boys called bacteria in your soil. Now it's stable. Now you can put heavy equipment on, it recovers. It really is resilient, it's open, it holds water. What happens when we till? We break it open. We chop up that white feathery material that's the fungi, we kill it, we break open the golf ball. The glue is a favorite food of the bacteria. They eat it, the macroaggregate collapses. So we have to stop breaking those macroaggregates to have the soil structure. And here's a good example. This is a picture not far from my home on the left, and I live in Northeast Indiana. These are some of the largest dirt clods I've ever seen. You could kill someone with it. They're bricks. There's no macroaggregates because it's been ripped so much that the soil is now all the small stuff that sticks together and clings. How is that going to hold water? How easy is that going to be to plant into? What kind of diesel is it going to take you to go across that field? Now look at the right. This is a customer who is showing off his earthworms, which again are a great sign of soil health. But look at where the sun is hitting that clod. All the little texture, those are the macroaggregates. Which one holds water? Which one is easier to work? Which one do you think has more nutrient cycling? It's easy. It's the one on the right. So we did a study. We looked at 44 farms across Iowa. We apply our product at planting. So it was applied late April, early May. And then we did different time points. We looked at bulk density and water infiltration. And it follows the bulk density decreases one application. Water infiltration increases over 50% as the roots and the fungi grow from where we applied and spread out. It's making more and more macroaggregates, opening the soil, holding more water. But we still till. Why? Well, one of the reasons is too much crop residue. So if you're not baling it off or feeding it off, which again, we want to return all those nutrients and that carbon food sources back into the soil, but we also know too much residue on top of the ground can make your soil warm unevenly. It can be later to warm. It can be a hassle when you're trying to plant through it. So you don't want massive residue mats in the spring. So let's get rid of that residue the natural way. So why is that residue? Why can a corn stalk, 1 or 2 years after harvest still puncture a tractor tire? It's because it's made of these things called cellulose and lignin. That's what makes a tree stand up to a high wind. They're basically, my analogy is cellulose is the rebar, lignin is the concrete. It's really hard to degrade. These are very, very strong compounds, as you see on the right, which I'm not going to go into any more detail. Fungi, though, Certain kinds of fungi, especially white and brown rot fungi, those are the best organisms on the planet that break down cellulose and lignin. And you can see that there in the upper left-hand corner, the fungi starting to grow on that corn stalk. So if we have the right fungi, by spring, you're not going to need to till because your residue is not going to be a problem. Our product, our breakdown product, is rich in these rot fungi, these saprophytic fungi, white rot, brown rot, etc. And this is a picture 10 days after application. Here's a field in Iowa, same field, just different parts, one with the product, one without. Everything was vertically tilled. Um, by the way, for any residue degrading product that's a microbial to work, we need temperature and moisture. These were great conditions. So if you're looking at your field and it's the middle of December, it's covered in snow and the ground is frozen, please do not spend your money on a microbial residue degrader. Wait till spring. But if you could put it down in the fall, when you still have some temperature and some moisture, that's going to do the job for you. Double cropping. Now, this isn't strictly a double crop. I've got pictures, though, from Tennessee and such, where they do double crop beans into wheat. This is a guy in Canada who planted his beans through standing cereal rye, and he applied our product over the top. This was following corn the year before, and his comments that look at how the rye is just degrading and you barely see any corn at all anymore. So we help enable even the double cropping without tilling, because remember, tilling really damages the soil. So I didn't talk much about what's probably most important to you. I've talked about reducing fertilizer and how biology will help build soil structure, help control residue. Healthy soil should give you higher yield, and we see it hugely. But healthy soil, you're actually making the ground better using less. And isn't the profitability what's most important? So how do you know if you're getting a product that has diverse biology? Well, our product is refrigerated. It's the only way I know to keep a living microbial, not spores, not dormant, not dead. Living biology, it's the only way to stabilize it. We make it, then we put it to sleep. It comes to you with an insulated wrap. You've got a good 8 weeks to use it, usually more. If it's sat out too long, it will tell you. You will not get your nose next to it. You just call us, we replace it, no questions asked. So that's rare that happens. But if you're getting a product that's powdered or stored at room temperature, it can't have all of this biology. It can't, because most of the biology needs oxygen, and we can't stabilize it other than keeping it cold. Again, for agriculture, two main products. The one in the spring helps with nutrient use efficiency, germination, rooting. We have the residue control product, the breakdown in the fall. Easy to use, apply as close to planting as practical. It mixes well with other things. The fall as close to harvest is practical. Again, once winter has arrived, I'd wait till spring. So I gave you a lot, and there's so much more. I didn't even talk about how healthy soil protects your crop from pests, from diseases. Again, there's so much going on. But sticking to nutrients, we had more guys this fall, literally, that were repeat customers that told us kind of sheepishly, well, I finally listened to you and I stopped using as much fertilizer. I had my best crop ever. And I saved more money on the front end. Think of what that does to your profit per acre, especially given the price of phosphate now, fertilizer in general, and low commodity prices. Again, do you want to be on team fertilizer, which is fine if that's what you want to do, or team biology? Let the biology work for you. When you use less fertilizer, the guys who have gone to no fertilizer or very low, do that stepwise. This is a tool that will help you dial in your production what the right fertilizer level is. There is no formula because your soil is different. Your soil is different across any one field, let alone field to field or other places where I could do trials. It's a tool. Move toward removal rates. For nitrogen, depending how bold you are, 10% cuts. If you're at a pound of nitrogen or more, there's a half a pound of nitrogen in a bushel of corn. You can back down. You can be more aggressive with P and K, but go to what's palatable and then see how it does. And next year, do another little cut. Use it as a tool. In 3 to 4 years, your fertilizer bill is going to look a lot different, but your top line revenue is going to look the same or higher. And really, really, really go as minimal tillage as you can. If you've got severe stratification, severe compaction, sometimes you're going to have to go and work the ground. But letting the biology do it to control the residue is going to be so much better for your ground. Because again, when you till, you kill fungi, you're destroying soil structure, so you're going to make compaction actually worse. And your number one source of nutrient loss is erosion. So you're going to have more erosion. And if you're worried about carbon, and you should, whether there's someone willing to pay you for carbon or not, carbon is the fuel in your ground. You lose a lot of carbon when you till. So these are our recommendations. And I hope I've given you enough of the science that convinces you that you can do this. And it doesn't have to cost you. It doesn't have to be a big risk for you. That it's something you can start moving toward. You don't have to go cold turkey, but this is a tool where 3 years from now, I don't think you'll recognize your farm in a really good way. So I hope I've been helpful, and I'm happy to take any questions, Ritu, if there are any. Oh, okay, so let me talk a little bit more about soil structure here. So I know I've had guys ask me what happens, um, you know, I, when I don't till, can I really take the risk? So I remember here, so I'm in Fort Wayne, Indiana. I met with a couple dozen growers, and they were just convinced they have to till or the compaction will kill their yield. And it's a 5-year road to profitability going to no-till, except for one guy in the corner who didn't say anything. And they looked at him and said, well, what do you think? You've been no-till. And he said, guys, it's not 5 years. It can be year one. The field I showed you in Iowa, in terms of the guy who used our breakdown product, he didn't till, he actually sold his chisel plow. There's another set of pictures where he's applying anhydrous in the fall. And by the way, I'm not a huge fan of anhydrous because it kills everything it touches. But I also understand why you use it because of its cost. If you're using it in the fall, you know, that's your operation decision. But in any event, he said he used 30% less diesel because the ground was so open. The rows closed easily above it. His neighbor literally across the road had to stop because his ground was so rough. He was losing too much of the ammonia because of smell. I mean, there's no mistaking it because his rows weren't closing. So it's all these little things that having better soil and better soil structure will do for you for profitability. So if there's no other questions, then Ritu, we have this as a recording, and I just want to thank you all for your attention. I hope that you found this interesting, and you can feel free to contact me or Holganix if you've got anything in here you want me to dive deeper on. Okay, so thanks a lot. You guys have a great day.